the Medical AI Podcast.
Welcome to the Medical AI Podcast with me, Dr. Felix Beecher. Strap in for a 30-minute thrill ride of big ideas. Skin cancer is the most common type of cancer worldwide, affecting millions each year. Early detection is crucial for improving survival rates. If not detected early, it can spread and it can kill. But it's also a good example of where AI systems have immense potential. With me to discuss this area is Cody Simmons, co-founder and CEO of a leading company in this space, Dermasensor. Cody, a very, very warm welcome to the podcast.
Thank you for having me. Really looking forward to the discussion today.
Great. Okay, well, look, could you introduce yourself and explain a little bit about what you do and what Dermasensor is all about?
Sure. So a bit about the company at a high level. So Dermasensor, our focus for a decade now has really been to create tools or solutions for the millions of frontline healthcare providers that are not experts in assessing skin lesions, to really enable them to more effectively detect and manage skin cancer. And ultimately, like you said, with the goal of more prompt and timely detection to really improve patient care. So that's been our goal. We've been CE marked for a couple of years, and we just received FDA clearance early this year. So, very excited after a very long and expensive journey to get to this stage. We've actually completed 15 clinical studies over the last decade to finally have our device available in the U.S. market. So, in short, for the first time in the United States, patients can now walk into their doctor's office, think family doctor, internal medicine, gynecologist, and if they have a mole or skin growth they're concerned about, they can point to that mole and say, what do you think about this? Do you think there's a risk for skin cancer? Should I go see a dermatologist? And for the first time, that doctor can provide them with a quantitative risk for skin cancer. That's never before been possible.
We will get into those details, but why don't we lay down some context and talk about skin
Sure.
cancer in general? Could you give us an overview of how serious a problem skin cancer is in general?
Sure. Yeah, so skin cancer generally, like you said, it's the most common cancer in the world. Melanoma is actually one of the most common, and it's quickly increasing in incidence. It's expected to be the second most common cancer in the U.S. by 2040, and it's one of the deadliest. There's about 5.5 million skin cancers diagnosed in the U.S. every year right now, and tens of thousands of deaths from across all three common types of skin cancers. The opportunity, the exciting thing about uh, uh, the clinical care aspects of skin cancer is that it's effectively curable if it's detected early. So even melanoma, the most deadly, 99% of it is curable if it can be detected in the earliest stages. And so that's kind of been the longstanding public health opportunity is, can we equip just in the United States, there's 1 million physicians and obviously many hundreds of thousands more when you think through nurse practitioners and physician assistants that are often asked by patients, or they notice a mole that looks suspicious, should they refer it or not? They're by definition, they are not experts at that. They're not specialists like dermatologists. So if they're able to more effectively assess those lesions like a dermatologist, then there's tremendous improvements possible in correctly detecting and managing skin cancer with a lot of not just benefits to the patients, but a lot of potential savings in the healthcare system too. Currently, an average primary care doctor is doing about 30 to 50 referrals or biopsies for every one melanoma that is diagnosed. So their hit rate is only 2 to 3%, whereas dermatologists are like 5 times better than that.
Now, some people might naively think that skin cancer can be, in some sense, detected fairly early. In that, if you have a strange looking mole with ragged edges, or maybe it's big, or it's growing, or it's a weird color, then that's suspicious, and they can go to their doctor. They can get it addressed or excised pretty quickly. Now, what's wrong with that naive view?
Well, most primary care doctors, both in Europe and the US, don't regularly do biopsies. They're typically, like with many of the dozens of specialists, they're deciding, do I send this issue onto the specialist or address it myself? Most primary care doctors in the United States are supposed to manage themselves about 80% of the patients that show up in their office. And so, you hear that often from primary care doctors, like, of course, we don't just refer a patient every time they ask about a mole they're worried about. We do our job, we assess it ourselves, and we decide whether they need to go or not. So a lot of PCPs don't do biopsies, to your question about cutting out themselves. And so they're typically making a referral decision. And, that's what studies, our studies, but dozens of published studies show that they miss a fair amount of skin cancer. Actually, the range in literature is anywhere between 12 to 46% of skin cancer that presents to them. They choose not to refer a biopsy, but they're also doing a tremendous amount of unnecessary referrals and biopsies. So there's a real opportunity to kind of improve the detection and the efficiency of skin cancer detection and management by improving primary care physicians' ability to do that.
Now, when we think about AI systems for skin cancer, I think, was a very early area for medical AI systems. Is that right? It's kind of like one of the cornerstones of
It,
early AI.
it was, yeah. The first publication was actually 1990 on applying algorithms to images of skin lesions. So it's actually been studied for well over 30 years. Um, And there's been various modalities, different technologies that have approached that. My background's more technical. A business background, but also bioengineering. I was in a biomaterials lab at Stanford for my graduate research, and um, types of spectroscopy, various types of imaging modalities like hyperspectral imaging, confocal microscopy, dermoscopic imaging, clinical imaging. So there's been AI, used to be computer vision, and then, the term has kind of changed over the years, but various kind of algorithms over the years applied to various data gathered from skin lesions. So yes, it's been studied, and there's been dozens and dozens of companies and hundreds of researchers that have worked on this. Actually, Memorial Sloan Kettering every year, I think the competition goes live in like a month, has research groups and companies submit algorithms. They'll make public, hey, here's the challenge this year, differentiating this cancer from that cancer, or psoriasis from acne. They'll kind of have different challenges, and groups submit like the best AI algorithm they have, and then they, let everyone know who the winners in the rankings. And there's hundreds of submissions. The last one they did, I think there was like 200 or 300 submissions.
So, given that it's a particularly active area for AI development, is there something about skin cancer which lends itself particularly well to machine learning kind of algorithms?
Well, I think most of because the widespread interest, and there's also many publications in it, is because physicians, especially dermatologists, capture photos as part of their standard of care. They'll take image of many dermatologists when they see a suspicious lesion. Before they do a biopsy, they photograph it. So there's these publicly available massive data sets. I mean, millions of lesion photos now that are available for skin lesions. So it's a natural, for technical folks, AI researchers, data scientists. It's, like I said, there's these competitions on it, right? Even so, for companies and researchers, it's a natural area to investigate and to use to push forward some of the boundaries of AI. The issue is with photos or images in particular. There's a very checkered past of those actually being effective in real world care, because the appearance of lesions can be so variable. And especially, there's been a lot of presentations at the dermatology conferences on how skin of color, different ethnicities poses a large challenge, and so there's health equity issues because most of the algorithms, like most doctors, are trained on what are more common visual appearances of these skin conditions, which is skin cancer is much more frequent on lighter skinned, on Caucasian patients. So that, if there's hair in the way, you have to standardize distance and lighting, and using the exact same camera type. If you switch camera types, then the algorithm will perform slightly differently. So there's, it's a great research project for tons to look into. Developing a commercial product that can actually prove in large prospective studies it's effective across diverse populations and work with regulatory agencies, that's a very different story. There's actually only two that are available in the United States right now. And interestingly, both use spectroscopy. There's none available in the United States that use any kind of imaging to provide any form of risk score or classification of skin lesions.
And given that, firstly, skin images are readily available in large numbers, but also the images themselves are probably less heavy in terms of storage than, for example, mammograms. And I guess that does that also mean that it's been a particularly thriving area for machine learning approaches?
Perhaps, but that's also, it's funny. We think of photos as pretty large, actually, because
Yeah.
we use
Okay.
a type of spectroscopy that's just zeros and ones. So our algorithm sits locally and processes locally. We don't need any cloud connectivity or anything. So images are definitely larger than that. But I see what you're saying. Compared to some other uh, medical tools and modalities, maybe they're relatively small. Yeah, I that's certainly helpful with some of the experimentation. But the downside, Felix, of photos, and this is, I'm technical enough to be dangerous, but I'm not really a technical person. I've never I was trained as an engineer, but I've never been an engineer professionally. And but what the real engineers tell me is the issue with photos is it's two-dimensional data, and so there's potentially hundreds of biomarkers in every single photo. So it's very easy to develop an algorithm that retrospectively performs very well on large data sets, right? But good luck locking that algorithm and then going and doing a new prospective study with dozens of new doctors and hundreds or thousands of new patients and having consistent, robust performance across all of them. Because whatever data set and whatever camera type and you trained it on might be slightly different shading, coloring, distance, patients with different skin types. Like there's so many variables in 2D data that it's easier. It's called, overfitting is the technical term, right? There's overfitting of the algorithm, and thus there's less generalizability of those algorithms and tools to diverse patient populations, which obviously Europe, America is diverse patient populations.
So, on the one hand, we have an area, skin cancer detection, we have an area which has been around for a long time in AI terms. And on the other hand, we have a very high prevalence of skin cancers. So, putting those together, you might think, it might be tempting to think that AI systems for detecting skin cancers is like everywhere. It's being deployed very commonly. Is that the case? Or can you put some numbers on the degree of deployment?
Yeah, no, it's kind of in the early days in the US. Do you want the answer for the US or Europe? Because they're actually quite different.
Both in that
I
case.
picked both. All right, I'll do a summary. So Europe, I'll try not to get too in the weeds. So Europe just switched regulatory structures from what's called MDD to MDR. So MDD, medical device directive, to medical device regulation, I think. MDD classified automated AI, skin cancer tools as the lowest risk category, the same as like a medical lamp. Because how they thought about it is, okay, it's it doesn't it's not interventional, it doesn't pierce the skin, it doesn't, it's for all intents and purposes from a mechanically, it's a camera, for example, or ours is a little handheld device that flashes light out of a tip, like not dangerous. But the new regulations are much more aligned with the FDA, and that, well, the device itself may not be harmful, but what the device is used for is a pretty high risk use. You're impacting healthcare professionals or patients' decisions on whether something is a deadly cancer or not, and whether it should be further evaluated and diagnosed or not. And that is a pretty high risk medical device. So the FDA has always had that stance. Europe has changed. So there were, I don't know, half dozen tools available in Europe. One called Skin Vision, for example, has been available for several years in Europe. I think has over 1 million users for patient home use, right? Um, but in the US, there's no image-based tools available now. And so there's a spectroscopy-based device called Nevasense that's been approved since 2017. They've had pretty limited adoption by dermatology. It's just for dermatologist use and just for melanoma. My guess, maybe like 50 or 100 clinics. And in the US, we just started being introduced 5 months ago, and we're already in use by a couple hundred doctors. So that's kind of the scale in the US versus Europe. So Europe, it's a broad mix of consumer use and healthcare professional use tools. Some of those are probably going to go away now with the more stringent regulations. We're actually in the process of complying with the new higher regulations that Europe has just changed to with the CE
I
mark.
see. So on the one hand, you might see that as disappointing. On the other hand, you might see that as an opportunity to squeeze out smaller competition. Is that true?
Yeah, I mean, honestly, the engineer and I guess human or member of humanity in me thinks it's a good thing. I guess the biased Dermasensor executive in me thinks it's a good thing, I guess, as well. But because, so for example, I'll just use unbiased data here. Memorial Sloan Kettering just published a paper maybe a year ago. They did a study, so they uploaded hundreds of their own diagnosed lesions that they use standard imaging systems for into, I think it was like 15 or 20 apps that provide some likelihood or scale for skin cancer. Some are available illegally on the App Store in the US, and Apple, I've heard, is in the process of pulling those down, and the FDA is cracking down on that. They actually the federal government fined and pulled some of them a few years ago, but they kind of bubble back up over time. And, um, but so Memorial Sloan Kettering did, uploaded hundreds of diagnosed images to like 15 or 20 of these different apps, mostly in Europe, some in the US. The average sensitivity was 40% for melanoma. So that means if the lesion is melanoma, that the algorithm correctly only said it was melanoma, this is high risk, or whatever the scale was. They defined that in the paper 40% of the time. And about half of the tools didn't even reach statistical significance. So it was like, coin flip or the roll of the dice in terms of what the result was. So that's what's pretty scary about a lot of these tools that where they're not highly regulated like Europe has been to date, or they just flout regulations and just try to get listed on an app store and have patients download them anyway. It's pretty scary, as Memorial Sloan Kettering showed.
Sure. Okay. Very interesting. So let's talk about Dermasensor because
Sure.
Dermasensor is in the very nice position of being considered to be a breakthrough product by the FDA. Could you just first of all tell us what Dermasensor is? Talk about the hardware and the software, and then after that, maybe talk about the process of getting recognition by the FDA.
So the hardware aspect of the device, we actually started with a 30-pound microwave-sized device that we developed in collaboration with Boston University and another group in Boston. So those two large prototypes, actually Professor Irving Biggio, a professor at Boston University, is the inventor of this field of spectroscopy called elastic scattering spectroscopy. It's also known as diffuse reflectance spectroscopy. So a lot of the early days, I put on my old engineering mindset and was in the lab with him and some of the other engineers at Boston, really understanding how the technology worked, and brought on, soon after I joined Dermasensor, our COO joined, Ryan Frieden, also, has a good technical mind, and started working with BU and other engineers thinking through how can we miniaturize this 30-pound, $30,000 system into a tool that our target users would actually use. Primary care physicians, for example, are not going to buy $100,000. So, if it cost us that much to make, we'd have to sell for a profit. they're not going to spend anywhere near that. And the system needed two people to operate it, one person at the touchscreen at like the computer interface on the device, and the other holding the probe tip, which was a long cord. So the early days are, okay, what's the engineering path to turn this 30-pound device into a handheld 1-pound device that our key design criteria were can be held and operated with one hand, is wireless, touchscreen, and the entire device use process from when you pick it up to when you get a result is less than 30 seconds. So that was kind of because the average consultation time for primary care doctor is about 10 minutes. So a lot of our physician collaborators and advisors are like, if this even adds a couple minutes to visit, it could get really tough for doctors to adopt this. Because, 2 minutes to 10 minutes, that's 20% added time to that visit. So that was really the early years, was miniaturizing that hardware. The two main components, there's over 400 components, most of which are relate to the printed circuit boards, the PCBs. But the two main components in terms of cost and kind of size of the device is a microspectrometer and a xenon lamp. So the broadband xenon lamp emits hundreds of different wavelengths of light, mostly in the visible spectrum. The spectrum is about 350 to 850 nanometers. So there's a 400 micron fiber emitting the light. There's a 200 micron fiber that detects the light. And then the microspectrometer, so is the light emittance source and the light detectant source. So the spectrometer analyzes, okay, of these hundreds of wavelengths that went out, how many light particles or photons came back at each one of those wavelengths. And then the algorithm runs and analyzes the 47 that, over the thousands of diagnosed lesions we've enrolled over those several years, are most indicative of malignant tissue. So the different light can pick up on when we pulse. I'll show you the device here in a minute. When you pulse the light into the skin tissue, it can detect things like nucleus size, melanocyte density, presence of condensed chromatin. So when you think about it, Felix, when a pathologist looks under a microscope at a tissue sample or a biopsy sample of a lesion, they're looking for those same things. They're looking, are the nuclei enlarged? Are there odd clusters of cells? And, those kind of cellular and subcellular features reflect light differently to the eyeball of the pathologist under the microscope. That's how human vision works, right? Those same features also reflect light differently to the spectrometer that you're holding in your hand in the device. So that's the basic principle of the technology, and also why the optics literature shows, and all of our studies that we've run show, there's no performance difference across different skin types, because we're not taking photos of the skin. Like, we don't, the technology doesn't care what the skin lesion looks like. It's assessing the cellular and subcellular level components. And we've shown, and the technical literature shows that background melanin content is not one of the factors for impacting the spectral signatures that the algorithm's, assessing for cancer.
Sure. So these kind of data that you're collecting with this device are fundamentally different from what would normally be collected from a dermatoscope, standard dermatoscope. Is that correct?
Oh, completely different. Yeah, it's deep into the surface of the skin,
Yeah.
but it's not even a photo. It's literally just measuring how many photons come back into the
Yeah, I
device
see.
at different wavelengths of light. That's it.
I see. Now, some people will be thinking, especially people who are in love with their smartphones, will be thinking, there's a very high quality camera on my smartphone. Maybe it's even approaching the quality of a dermatoscope. And they might also know that there are apps on app stores which claim to be able to determine what kind of mole you have and whether it's cancerous or not, and whether you should go and see a dermatologist. Now, so presumably there is some sort of added value for your system on these kind of free phone apps. Could you just unpack that and describe that a little?
Yeah, I mean, the main one is that our device is actually accurate and consistent, right? So that's the Memorial Sloan Kettering study I was mentioned, which was pretty recent. I think it was published a year and a half ago. Evaluated a lot of those apps you're describing. That none are available in terms of receiving FDA authorization in the U.S., but some are listed on the App Store anyway, which is scary. And then some are actually appropriately available in Europe. And the Memorial Sloan Kettering showed the average sensitivity of these apps to melanoma is 40%. So if you take a photo using your, the standard of the art algorithms running on the tools and your great smartphone, and you take a photo across all these apps, the average rate for telling the user, hey, this is concerning or this is likely to be melanoma, is only 40%. So that's what's really scary about, in theory, you would think, oh, smartphone, you have good photos, these should perform well. But unfortunately, in practice, they don't. And that's actually the only one that I recall that really stood out in terms of performing pretty well. It wasn't great, but pretty well. Was the one, Skin Vision, that's in Europe, that's been available to hundreds of thousands of patients. So credit to them that they were clearly above the pack in terms of the performance level. I know they've been trying to get FDA clearance for several years, but the FDA has, I think, legitimate concerns with patients' self-use. So, for example, okay, if you have a mole on your arm, you take a photo of the mole, your smartphone, the app confirms it's benign. What if you have melanoma on your back? So now you're not going to the doctor, right? Like, you're not going to the doctor because, oh, my arm's fine. It's like, yeah, but then you can die 2 years later because you didn't have anyone evaluate you, right? And that's what doctors say. A lot of times patients, they think this mole's fine. They get their shirt off, they evaluate them. They're like, yeah, but this one's very concerning, right? So patients do a pretty good job at presenting suspicious lesions. Studies show actually about 50 to 70% of melanomas are initially presented and identified by the patient. But, that leaves 30 to 50% that patients didn't identify, didn't know about, right? So I think that's FDA's big concern. If it was just your eyes, for example, you only have two eyeballs. Everyone can use a tool to analyze two eyeballs, for example. But a mole, you could have one, you could have 50. Like, how do you make sure patients are effectively using that at home? Even if the technology performed very well, how do you know, are they actually using it on the suspicious lesions? It's very hard. And I mean, I think it'll happen in our lifetime, but with the current regulatory paradigm and performance of tools that can be easily used by patients at home, not in the next 5-10 years. I'm quite confident in saying, having just gone through the FDA process. Yeah.
Sure. Okay. All right. Well, last question, and it relates, I think, to what you were just saying, and that's a question of how systems for skin cancer detection perform differently depending on skin tone, like light skin versus dark skin. So, could you talk about, firstly, what the kind of differences are in detection rates, but also, I think part of the context here that's important is whether skin cancer rates themselves are different comparing people with light and dark skin.
Sure. Yeah. So I mean, there's definitely um, the incidence rate of all skin cancers, melanoma, basal cell carcinoma, squamous cell carcinoma, are higher in lighter skin patients. The scale uses Fitzpatrick skin type 1 to 6. And it's defined as, part the color or tone of the skin, but also things like, what is your hair and eye color? Do you burn? How easily do you burn? Do you burn? Right. So Fitzpatrick skin type 6 basically cannot burn. Dark skin type cannot burn. Dark hair. So on that, typically African American in the U.S. African American, but can still get melanoma. For example, Bob Marley died of melanoma. The incidence of it is much lower, but they've actually shown melanoma is more deadly in patients with darker skin types, in part because it's not monitored and evaluated as well, because it is rare, and in part because it's often in places like, it's called acral skin on your hands or on your feet, that you're not looking for, and you're maybe more hesitant to do a biopsy because it's your hand. And so sadly, maybe there's also kind of health equity issues as well. I read in one paper that explains why the outcomes for patients diagnosed with melanoma is fairly different based on skin type and ethnicity. And so that's something that tools like ours and maybe other future tools could help with, is helping healthcare professionals more equitably assess suspicious lesions across all patient types, because most of their medical training is on the patients that that cancer is more common on, which is typically more lighter skinned Caucasian patients.
So have you done a head-to-head comparison with people with light skin and dark skin in terms of the sensitivity of your systems?
Yeah, we yes and no. So we had a very large sample size of lesions on darker skin types. So 1 to 3 is considered lighter, 4 to 6 considered darker, sometimes 5 to 6. Our pivotal study had over 1,000 patients, and 28% of those had Fitzpatrick skin type 4 to 6. So that's, about 280 patients. And I think Fitzpatrick skin type 5 to 6, I think it was like 12% or so. So still, well over 100 patients. However, and so that was really encouraging, I think, to the FDA. They had us do a lot of subgroup analyses comparing different skin types. We even looked at, lesions that are the same pathology, like a seborrheic keratosis or nevus, but have different pigmentation levels. Does that impact performance? Because that's another indicator, right? Is melanin and, the dark, the lighter dark nature of the lesion changing things, or the patient. So we really didn't see any meaningful performance differences. So we're happy to see FDA that there's no restrictions on device use based on skin of color. However, they do understandably have a note. It's one of the precautions saying that there weren't a lot of cancers diagnosed in the darker skin types. So yes, there was hundreds of lesions enrolled, but the large majority were benign. So there weren't many cancers. I think it was like 15 of the 224 were diagnosed in the darker skin types. So to rely more on clinical judgment for those darker skin types. And accordingly, we're doing what's called post-market surveillance, gathering additional data with a focus on darker skin types to further add to that data set. So that was part of like the agreement with FDA was, the technology inherently how it operates and the evidence suggests there's no meaningful differences. Like sensitivity and specificity and accuracy was all within a few percent of each other for lighter and darker. But the sample sizes weren't very large for the cancers in darker skin types. So let's keep monitoring that in real world use to assure that. So that, which we thought it's expensive, investors weren't too excited about it. But I think, for patient population and from the FDA standpoint, it was reasonable. And Felix, they did raise a couple times that they do not want to happen with this new category that the approval of our device created. They don't want that to happen in this area. What happened with pulse oximetry, for example, pulse ox, in which recently it was found that pulse ox is actually not very accurate at all in African American patients, which I think it's been. I mean, you're a physician. I'm not. I think it's been widely used for like decades, hasn't it? Some of these pulse ox tools. Yeah, so it's a pretty scary thing to find out decades. To find out decades later, I laugh so I don't cry. Yeah, so they wanted to make sure, these new AI tools, even though our technology and our clinical studies suggest there's no meaningful difference, let's make sure and let's keep monitoring that performance out in the real world.
Well, Cody, it's an extremely important area of medical AI, and it's been a brilliant discussion. I'm very much obliged to you, so thank you very much indeed, and best of luck with Dermasensor.
Thank you, Felix. I appreciate it.
Great. Cheers. Thanks for listening. Ping me on LinkedIn if you have any ideas on new subjects or guests.